Hey everyone, it's Dr. Fox. Hope you're doing well. Let's talk about blood type. In this video, we'll discuss the nature of blood groups and how they're determined. We'll also discuss the ABO blood group and the RH blood group and some of their ramifications. So blood types are ways in which we can categorize and describe meaningful interactions between different types of blood. Now, blood cells, like other cells, have surface molecules on them. And so we can use these surface molecules to begin to categorize them. When we look at the surface of an erythrocyte, there are many different proteins and glycosphingolipids and carbohydrates, etc. And each of these are capable of perhaps having an immune reaction mounted against it in a foreign environment. And so we call them surface antigens, or sometimes agglutinogens. And that's because oftentimes antibodies will grab a hold of two of the same and begin to agglutinate or clump those cells. Now, anytime we bring an antibody and an antigen that it can respond to together, we will have an immune response. And that immune response might be agglutination. It may also be a complement response, whereby the antigen and anything in its vicinity are destroyed by the immune system. So there's a variety of ways that the immune system can deal with potential pathogens, i.e. Antigens, by means of antibodies. Now, what's interesting with respect to blood types is there are over 100 genetically identified blood types and about 35 recognized systems. Now, we are going to discuss two of the more common systems that are important for cross-matching, the ABO blood group and the RH group. But there are many more than these which are very clinically relevant. So let's start with the ABO blood group. The ABO blood group is named, or its subtypes are named by their surface antigens. Now, these are going to be glycosphingolipids, and so we say there is a type A antigen and a type B antigen. And so a cell may have either, and if it has only type A, then it is ABO type A. If they only have type B, then they are type B. If they have both, then they are type AB. And if they have neither the A or the B antigen, we say they are type O. Originally, this was meant to be 0, but we just call it O. Now, one of the more interesting things about the ABO blood group is that it's very typical for a person to have antibodies against the surface antigen that they don't have on their own red blood cells. So, for instance, someone who is type A will have anti-B antibodies, specifically IgM, which are agglutinators within their blood plasma. Someone who is type B will have anti-A antibodies. Someone who is type AB will not have any of these anti-A or anti-B antibodies. And someone who is type O will have both anti-A and anti-B antibodies within them. So if we think about how this might play out, someone who is type A, if they receive whole blood from someone who is type B, then what happens is their anti-B antibodies will agglutinate that, causing a potentially lethal combination. Likewise, someone who is type B, if they receive blood from someone who is type A, will also have an agglutination response. Someone who is type AB can receive blood from someone who is A, B, AB, or O with no agglutination. And so we say that someone who has type AB is the universal recipient. They can receive blood from anyone. Now let's look at someone who has type O. Recall they have both anti-A and anti-B antibodies. So if they receive blood from someone who's type A, B, or AB, that's not going to work out well for them. So someone who is type O can only receive blood from someone else who is type O. However, because the O has 0 antigens on the surface of the red blood cells, they're capable of donating blood to any of the other blood types. And as a consequence of that, we say that someone who is type O is the universal donor. Now, when we talk about transfusions, we were just talking about that in the context of whole blood transfusions, which is really exceedingly rare and probably only done outside of modern hospitals in times of extreme emergency. These days, when there's a blood transfusion, people receive either blood, which really is just spun down or packed erythrocytes, or fresh frozen plasma. Before doing any sort of transfusion, however, oftentimes, and this is especially important in terms of blood type of doing or a whole blood transfusion, doing a cross-match test. And that's when you would take some red blood cells from the donor, mix it with a recipient plasma sample, and if there is no agglutination, then it is safe to transfuse. If there is agglutination, it is not safe to transfuse. Now let's talk a little bit about the Rh factor. The Rh antigens are integral membrane proteins, so they're not glycosphingolipids, but rather proteins within the membranes of the erythrocyte. And what happens with the Rh factor is that a person can either be Rh positive or Rh negative. Someone who is Rh positive has the Rh factor. Someone who is Rh negative does not have this integral membrane protein. But they have the capacity for making anti-Rh antibodies. And so what can happen, and where this is most clinically relevant, is in a scenario called hemolytic disease of the newborn. Now imagine this. Imagine we have an Rh negative mother with an Rh positive fetus. The mother and fetus are connected by means of the placenta. Typically, IgM antibodies are too large to cross the placenta, but there are IgG antibodies which are very small. And so if during the process of gestation there's any exchange of blood product across the placenta, because maternal and fetal blood don't typically mix, but there can be examples of fetal blood crossing the placental barrier, then an Rh negative mother will begin to mount an immune response against the Rh factor. And those IgG antibodies can cross the placenta and they can attack the fetus. This is most common with subsequent pregnancies, but it can affect a first pregnancy. And so when you have an Rh positive fetus in an Rh negative mother, you run the risk of both hemolysis and agglutination, complements of the IgG antibody, which crosses the placenta. Now, there is a way of preventing this, and it's a rather simple injection. It's something called RhoGAM, And for those individuals who are Rh negative, during their first pregnancy, RhoGAM is indicated prophylactically to just ensure that there isn't an immune response. It's a very economical and effective way of preventing hemolytic disease at the newborn. Now let's assess our knowledge. For which blood type would be the easiest to find a transfusion donor? Type A, type AB, type B, or type O? Well, we know that type AB is the universal recipient, and so they can receive blood from type A, type B, or type O. Therefore, type AB would be the easiest to find a transfusion donor. Thank you so much for your time, and I look forward to talking to you in the next video.